Welcome to this presentation on the gut microbiome entitled Studying the Gut Microbiome, How We Got Here, What's Happening Now, and Where We're Headed. My name is Tyson Dawson. I'm a PhD student at the George Washington University, and I study biomedical sciences, genomics and bioinformatics specifically. And today we're going to talk a little bit about the history of gut microbiome research, what's happening now in gut microbiome research, and hopefully we can get a glimpse as to the developments that are soon to be in store for this field of research. So let's get started. As we begin this presentation, I want you to think about why researchers would want to study the bacteria in the gut, and hopefully over the next several slides I can start to answer that question. Let's begin a few hundred years ago with Leeuwenhoek, the father of microbiology. He was the first to report on these tiny little organisms that existed in and on the human body. He actually observed bacteria in saliva samples through his sort of primitive microscope, and he was quoted as, he wrote that animalcules, which are what he called these little organisms that he saw, animalcules were in such enormous numbers that all the water seemed to be alive. When he was looking at the saliva of these old men that he was, observing in the microscope, he saw these tiny little animals and reported on them. And since this discovery, there have been, decades of research into what exactly these little animalcules are. Today we would call those microbes. What these are and, what role they play in health and in disease. Some of the earlier research going back about 80 years ago in the 1940s began with what are called germ-free animals. I have a quote here on the slide that defines what a germ-free animal is. The term germ-free or a xenic refers to an animal demonstrably free from microbes, including bacteria, viruses, fungi, protozoa, and parasites throughout its lifetime. So these are mice usually, but there can be other animals as well that are bred in such a way and they're kept in an environment in which they don't have any microbes in or on their body, and as such it allows us to study what role microbes have, and specifically the absence of microbes. One thing that's especially valuable is that it allows for monocolonization, is this word here. So you can colonize for example, the gut of a mouse, for example, with just a single bacterial species and study what effect that bacterial species has. So it kind of allows you to control your variables instead of having all of these different bacteria to worry about and not being able to isolate what the particular effect of a single bacterial species might be. You can monocolonize and just have one bacterial species, or you can have a gnotobiotic mouse, which is one in which every microorganism present is defined. So that could be one or more, maybe you have one bacterial species or two or three, but you limit the number and it only has the bacteria or the microorganisms that you want them to have and that you want to study. So that's germ-free animals, which are really useful. And some of the early discoveries in the world of microbiome science, the gut microbiome and other science studies, other disciplines related to microbiology and these animals. This slide details some of those original findings, and I want to especially make note of at least a couple of these. This point is especially interesting, that these animals have a need for a higher caloric intake to maintain their body weight. I think the germ-free mice can eat about 30% more calories compared to their conventionally raised counterparts. And this is likely due, as it indicates here, due to less efficient whole body metabolism. So these animals don't have these microbes that help them digest their food, and as a consequence, they need to eat more calories in order to stay alive. Another interesting point that I want you to focus on is this last one here, that indicates that some germ-free animals, most germ-free animals, lack important metabolites, and in mice in particular, this indole-3 propionic acid has been studied, and it turns out that it doesn't exist, this metabolite doesn't exist in germ-free animals, But if you monocolonize the animals, so you just introduce one bacteria to them, and in this case that bacterium is Clostridium sporogenes, if you introduce that bacteria into their gut, lo and behold, they start producing that metabolite again. And further studies have indicated that indeed it seems like Clostridium sporogenes has a gene which allows them to act upon specific substrates in the gut and produce this metabolite that's important for mammalian health. And so we'll get more into this field later, but this is where the world of microbiology and the world specifically of the gut microbiome comes in contact with a discipline called metabolomics, where you're studying the metabolites of a particular sample or of a particular organism. And it turns out that these microbes are very important in terms of creating these metabolites that are often essential for life as we know it, certainly as humans. Some other important studies, early studies in the world of the microbiome. This one happened more recently than some of the last studies that I mentioned, but studies on human twins and fecal transplants. This study is especially interesting and has a lot of ramifications in the world of the gut microbiome. It turns out that if you have a set of twins, these are human twins, that are discordant for obesity, meaning that one of the twins is lean and the other is obese, and you take fecal samples from each of them and you introduce those fecal samples as a fecal transplant into mice, it turns out that the mice who get the fecal material from the obese twin will gain weight in a significant amount. And we'll view a chart in the next slide. But the major findings from this study, I'll point to this point here, that there's evidence that a donor's community will be replicated following a fecal transplant. So if you have a particular composition of microbes in your stool, for example, you can transfer that same community into a receptor organism, into whoever is the recipient, and that will point to this point as well, that there's evidence for the transmissible and modifiable interactions between diet and microbiota influence host biology. So you can transfer fecal material and it'll influence the recipient's biological processes. And we have a graph here that indicates that. In figures C and D, they're comparing the recipients of the lean twins fecal matter, and then the mouse that received the obese twins fecal matter. And as we see, the mice that received the obese twins fecal matter saw a drastic change in both body composition and in fat mass. So really interesting results that you could see phenotypic changes following a fecal microbiota transplant. This quote here from the paper I thought was especially interesting. It says, "The intact, uncultured, and culturable bacterial component of obese co-twins' fecal microbiota conveyed significantly greater increases in body mass and adipodicity than those of lean communities. Differences in body composition were correlated with differences in fermentation, metabolism, and microbial transformation of metabolites, as well as receptor signaling." So not only were they able to say that receiving the obese twins' fecal material in a fecal transplant changed the weight, they were able to describe why exactly that happened, and it turns out that it was because of the metabolite changes on the level. It happened because of the fermentation changes, the metabolism changes, that these microbes normally transform the metabolites, and for whatever reason that happened in a different way in the microbial community of the obese twin, and that was replicated in the mouse that received that fecal transplant. So this is all very interesting, and it helps us answer one of the core questions of biology that has emerged, especially since the Human Genome Project was completed. The Human Genome Project was completed almost 20 years ago, but prior to that, many researchers thought that the human genome would have on the order of maybe 100,000 genes. And that estimate was based on some biochemistry, and it also sort of just made sense, based on some math. They said, okay, well, humans must have a lot of genes because we see so much phenotypic difference between people, And a large number of genes would help us explain all the different diseases that we have and just the complexity of the human body. Obviously, as researchers, as humans, we like to think that we're kind of at the top of this evolutionary chain, that we're super complex. But it turns out that we only have 20,000 to 25,000 genes, which is the same number of genes as sort of what we view as lower organisms. So that was a surprising finding, and following the completion of the Human Genome Project, we also weren't able, obviously, to cure all diseases. It wasn't this grand revelation that all of a sudden we could fix everything. It turns out that there are still mysteries of the human body that can't be explained by our genetic code alone. And so some of these diseases and states that have sort of escaped our understanding might be explainable by our gut microbiome and maybe not by our actual, our genomic information encoded in our DNA. And that has led some researchers to claim that our gut microbiome the human microbiome in general, could be considered a second genome. This is a quote from a paper entitled "The Human Microbiome, Our Second Genome." And in that paper, they describe the human microbiome as an essential but largely ignored overlay to our genomic blueprint. It is a source of genetic diversity, a modifier of disease, an essential component of immunity, and a functional entity that influences metabolism and modulates drug interactions. So, it turns out that as humans, we don't just have a set of instructions that code everything we're able to do. It turns out that we have these microbes that also have genes that are important, that we didn't have to evolve to have these genes, because the microbes within us are able to do these chemical conversions and undergo these metabolic processes that are important for our health, and that the information encoded in these microbes might have a lot to tell us about disease and immunity and metabolism and how drugs interact with our body, which has again led researchers to call this potentially it might be considered a second genome. Before we get into discussing this a little bit more, I think it might be important when we talk about the microbiome, what exactly it is that we mean. The microbiome is the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. So commensal organisms being those which don't benefit us but don't harm us. Symbiotic, those which we benefit because we're giving them a place to live and we're giving them substrates upon which to feed. And they are giving us something in return. Maybe they're transforming those substrates into something that's useful for us. And then the pathogenic bacteria, which are those which are harmful to us and exist within our bodies to our detriment. And many papers cite that up to 90% of the total number of cells associated with our bodies belong to our microbiome. That might sound like a crazy statistic, that the cells associated with you are weigh heavily in the favor of the microbes rather than your own cells. But this can be understood given that these prokaryotic cells are much smaller than your own eukaryotic cells. But it's still nothing to scoff at. You have within you right now pounds of microbes in and on your body, and so they play a very important role, even though we obviously can't see them with our naked eye. Now let's move on to how we study these bacteria. And one important note before we go forward is that current culturing techniques don't allow us to completely study and understand these bacteria ex vivo, outside of the human body. Culture techniques began in the late 1800s and were developed to grow and study bacteria. We learned a lot of interesting things about bacteria from this, and a lot of the foundational principles of bacteriology come from this era. But it turns out that the number and the diversity of cells that you can observe microscopically in a stool sample, for example, far exceed the number of cells that you can grow in culture. So if you take a stool sample and you look at it under a microscope, maybe you could observe 100 species, but if you were to put that same stool sample and try to culture the bacteria, you would get, maybe a dozen or a couple dozen, but certainly not 100 like you saw under the microscope. And this is because culture-based approaches introduce biases. Specifically, it biases those bacteria that are able to thrive under laboratory conditions and in isolation, whereas those that don't, that have a more specialized niche that need the specific pH or the specific temperature, the specific substrates available on the human body, will die, and they'll die before we can study them. And so the full diversity of microorganisms probably can't be explored by culture techniques alone. That doesn't mean that we're not trying. Some researchers are hoping that by developing more sophisticated media, using semi-permeable membranes, and using different isolation techniques, that we can be able to study a greater variety of bacteria in the laboratory. This is certainly possible, but I'm not sure if anyone believes that we'll ever be able to culture every single bacteria that lives in and on the human body in the laboratory. So another way that we could potentially assess the microbiome diversity that we see in the microscope would be to grow these bacteria sort of on a culture plate, but inside the body, or inside an environmental sample, or wherever these bacteria grow or live or thrive naturally. We could place a sort of scaffold for them to exist in that place naturally, culture them where they already are, and then take out that scaffolding and then observe them. And this is referred to as growing the bacteria, growing the microbes in situ, using, in this case, this diagram shown here is of these eye chips that have been developed to do this sort of thing. And so that's potentially a possibility, but at this point, these are too large to put inside of your body, certainly, and so it's not necessarily feasible for what we would like to do. So in the absence of being able to grow the bacteria, we could also use a surrogate, which will be the focus of the remainder of the lecture, which is genomic material. Microbes, just like you, have DNA, have genomes, and those genomes lend themselves to analysis. And there are two major ways in which we analyze bacteria or microbes using genomic material. There are two approaches to sequence that genomic material. And those approaches are 16S amplicon sequencing and whole genome shotgun metagenomics. And so we'll get into those in the later slides. But first, let's talk about how powerful it is to have this surrogate. There's an incredible information density in the microbiome. It's been reported that each teaspoon of stool contains in its bacterial DNA so much data that it would take 100,000 of today's highest capacity thumb drives to store all of that data. And this would be about 1,200 kilograms, or the weight of a young giraffe. So these are, this is a lot of information that's encoded in these bacteria. I think this particular estimate was made assuming that the largest thumb drive you could buy today is maybe about a terabyte. And so there's 100,000 terabytes of genomic information in just a single teaspoon of bacterial and just a teaspoon of stool. The information being encoded in that bacterial DNA. So by taking a stool sample or a sample of a swab of your skin or in your nose or in your mouth, you can certainly find a lot of bacterial DNA in there, and that bacterial DNA lends itself to analysis. And so let's talk about how we can analyze bacterial DNA, microbial DNA. So the first approach is amplicon-based sequencing. This is sort of the older approach, but it makes a lot of sense. Basically, in this type of approach, you amplify a single gene. In this case, it's this 16S rRNA gene. It's a 16S gene in microbes, and in fungi and in protozoa, it has analogs, which we'll get to later. But essentially, instead of looking at all of the genomic information, you are looking at a single gene. And this gene has been carefully selected and considered because it shows it shows enough similarity across across all of bacteria that it's well conserved. Every single bacterium has this particular gene, but it's also variable enough that you can differentiate between species based on this gene. So it's a really good candidate. Is what you do is you find these primers that can attach to the beginning and end of this 16S sequence, and then you, doing PCR, you amplify this region of their genome, this gene, you amplify it so that there's millions and millions of copies of it, and then you can sequence those, and when you're sequencing, this is metagenomic sequencing, so you're not just sequencing one organism's, the genes from one organism, but from many hundreds or thousands of organisms, and then it can give you, by comparing the sequences that you acquire to known sequences, you can get an estimate of what the population distribution is. If it's 17% bacterium A and 13% bacterium B and 14% bacterium C, it'll give you this this analysis of what percentage of your sample belongs to each taxa of microbe. Here's another schematic again you have these primer sequences and you're targeting this 16S gene that has a number of variable regions that will change depending on what species you're analyzing, and so each 16S gene is unique to its the species that it corresponds to. So that's 16S sequencing. We'll get into whole shotgun metagenomic sequencing here briefly, but let's first look at sort of an overview of why you would want to do microbiome research in this way and what sort of insights doing metagenomics and in terms of the microbiome, what sort of information can that give you? And there's sort of three levels of analysis. There's metagenomics, metatranscriptomics, and there's sort of the systems biology approach. So maybe it makes the most sense to start with metagenomics, which is looking at the DNA of a sample. By looking at the DNA of a sample, DNA is a more robust molecule, so it allows you to look at what microbes are present. Again, by you're sort of taking all of these puzzle pieces and putting them together until you have a picture of all of the different microbes that are present by looking at their DNA. And it allows you to maybe start to think about functional profiling, which is what role in the body might these microbes play? What sort of metabolic activity are they undergoing? Now, it doesn't give us as high of a resolution, but it is able to give us the taxonomic profile, the profiling of how many bacteria belong to this kingdom or to this phylum or to this class or this order or this genus or this family. It allows you to see how the bacteria are spread across this sort of tree of life and what weight each of those branches has in your sample. Metatranscriptomics is another type of analysis that you can do, and it allows you to look at the RNA of a sample, and so it allows you to see what sort of gene products are the microbes in your species actively transcribing. RNA is a less robust molecule, so it doesn't last as long in a sample, but that means that you can sort of get a snapshot of what they are doing, like this indicates here. What are the microbes doing? What sort of processes are they involved in? So it still allows you to do taxonomic profiling, but it allows you to do taxonomic profiling of those microbes which are active. And because DNA being a more robust molecule, you might pick up DNA from organisms that are in your sample but that are dead. And this would be, metatranscriptomics would allow you to do taxonomic profiling of bacteria that are still alive. It would also give you more definition in terms of the functional profiling that you're able to do, because you're looking at what sort of genes they're transcribing, and that gives you a better lens as to what sort of metabolic activity are these microbes undergoing and what sort of function might that have in the host. And if you put all of this together and you combine it with host transcriptomics as well, so you're not only seeing what are the microbes doing for the host, but what is the host doing in response to the microbes? If you add that in, then you have yourself a systems biology approach where you're looking at how one affects the other and this bidirectional relationship that everything has, and you kind of throw it all together and you can get a very interesting analysis. And this is maybe not quite as much of a reality as we would like it to be, but a lot of advancements are coming from this area of systems biology. So that's sort of what you would, what sort of the different approaches towards microbiome research allow you to get at. This is another depiction of some of the things that you can do with metagenomics and microbiome research. This particular figure is split up into two regions. This over here on the left, sort of this blue box, is what is associated with 16S rRNA gene profiling, which again is this sort of older method that we've discussed. And then on the right, we have what a shotgun approach allows you to do. We'll get into a shotgun approach a little bit later, but a shotgun approach essentially allows you to not only look at one gene, but look at every single gene that's involved. It allows you to look at the aggregate of all of the genomic material in your sample. So 16S rRNA gene profiling allows you to cluster organisms together in what are called operational taxonomic units, which are groups of organisms that are very similar to each other. And then you can assign those OTUs particular abundance values, and you can get a sense of what sort of taxa are belong to your sample. But shotgun approaches allow you to do something very similar, but in much more definition. So you're able to look at all of the DNA associated with your sample, and in a similar way you can map them not only just to operational taxonomic units, but to actual genomes and to actual genes. And it also allows you to, maybe you're sampling something and this bacterium has never been identified before, so it doesn't map to anything. You could possibly assemble an entirely new genome and discover something that you weren't even looking for. That's one of the downfalls and one of the drawbacks of 16S sequencing, is that you can only look for what you already have profiled before. You have to be able to compare this 16S sequence to something in a database, and if it doesn't get any hits, then you're not really sure what it comes from. You're able to partially overcome that, at least with a shotgun approach. Shotgun approaches also allow you to look at viromics, look at the viral particles. Viruses aren't probably going to have this 16S gene, but they do have some sort of genomic material, and with a shotgun approach you're able to capture that. And then metatranscriptomics, which again allows you to look at the RNA of a sample, is possible with shotgun approaches. We discussed this a little bit already, but it allows you to look at what is actively going on in the sample, what sort of pathways are active, and you could organize the species into pathways and see sort of what's going on and who's doing it. So that's all very fascinating. I promised that we would talk a little bit about a shotgun approach, which is sort of the newer technology that 16S sequencing is sort of being phased out in favor of shotgun sequencing by many researchers. And this has, let's go back, this has a lot in common with this particular technology utilizes often Illumina platforms, but could use sort of these third generation sequencing technologies like PacBio or nanopore sequencing. And essentially what you do is you extract some DNA or potentially RNA, you fragment that genomic information, and then you put it into a sequencing machine that has a variety of steps, and out comes a DNA sequence, and comes out all these different reads, and then it requires you to align those reads to see what genomes they correspond to, and that's detailed on this slide, where again you've sequenced and then you assemble those into contigs where you have these things that align to a particular genome. You figure out what taxa they belong to, and then you assign them to a particular genome, and then you can enumerate how many E. coli reads did you get in your across each sample? How many times did you see this E. coli, or how many times did you see this bacterium? How many of your samples didn't align to anything and you're not even sure what it is? And so this is very useful. Allows you to count the number of species or strains per sample. Again, this is going into the application of some of these things. So again, we've talked about you could do sample collection of a microbiome. In this case, looks like the oral microbiome. You could you extract the DNA. You can do some metagenomic sequencing, and then once you have those sequences, you could put that into a taxonomic profile, see what taxa they belong to, or you could put into a metabolic profiling pipeline. In this case, the example tool that they give here is this tool that's called Human. And you can see what sort of functional pathways these microorganisms are involved in, and it can give you some nice figures in terms of what's going on, what percent of your sample is involved in maybe carbohydrate metabolism, or different subcategories of different metabolic activities, and maybe what body sites they correspond to as well. You could find biomarkers. So this is, there's all sorts of different tools that once you have this metagenomic data, you can throw it at a tool and it can give you some really interesting results. This is going back to amplicon sequencing, which we discussed before. So again, we have two different types of approaches to metagenomics. You have amplicon sequencing and whole shotgun sequencing, whole genome shotgun sequencing. And this is just another review of amplicon sequencing, where you extract the DNA and you do PCR to amplify the genes of interest, assign those to operational taxonomic units, and then you have to use a database that has all of these different 16S sequences and what species they're associated with, like RDP or Silvo, which are these databases, and then it'll give you a similar counts table. So again, at the end of the day, you get something that's pretty similar, but whole genome shotgun metagenomics gives you a clearer picture of what's actually going on, even if it's maybe more expensive. And this is just an illustration. Again, we've kind of been talking about this a lot, so I'll just go over it briefly. But this is an image of the 16S rRNA, which is the gene that is being looked for in 16S amplicon sequencing, and it has these variable loops and conserved stems. So, and this chart sort of shows that there's at some regions of the genome there's very high sequence identity, meaning that it's common across the entire kingdom of bacteria, and in other places it's quite low. And so because you see this variability, it allows you to there's enough similarity across the sequences where you can align them easily and they're sort of universal, but at the same time there's enough diversity where it allows you to really be specific and be able to distinguish one species from another. This is another example of a biological question that you might be able to answer from microbial taxonomic profiling. You get this table of the different, in this case, phyla that are present in your sample. You can group those together, and then this particular chart shows this LDA score, which is associated with biomarker discovery. And so maybe there are particular taxa that are associated with a particular phenotype or a particular disease, and so you could identify those as biomarkers and say, if you have this particular microbe, it might be indicative that you have a susceptibility to a disease, or you're currently in a disease state. Maybe you have inflammatory bowel disease or something like that. So we've sort of talked and done a quick overview of all sorts of different things that you can do with the gut microbiome. And now let's talk about some large-scale projects that have happened in the past sort of 15 years in terms of microbiome science. The first of these is the Human Microbiome Project, which was an NIH initiative from 2008 to 2012. The Human Microbiome Project was sort of a natural outgrowth of the Human Genome Project. The Human Genome Project was completed in the early 2000s, And following its completion, we had all of these researchers and all of these machines, and people were just itching for something new to do. So they decided to identify and characterize all the microorganisms, including pathogens, living in healthy humans. This was maybe too much of an ambitious goal, because here in 2020, 11, 12 years later, we still don't fully understand what a healthy human microbiome looks like. But this was an important first step, and taught us a lot about what's really going on. They had 242 healthy individuals that were clinically screened for absence of disease, so they were known to be healthy, and they sampled from 5 body regions: the mouth, the nose, the gut, the skin, and the vagina, and 18 body sites. And so, within each body region, they took maybe multiple samples, and then they had a couple visits to the clinic, and we got some metadata from them. And they used primarily 16S rRNA amplicon sequencing, which is sort of this older method that we discussed before, but some of the samples were subjected to shotgun sequencing. And so let's look at some insights here. We won't go into all the details of these figures, but essentially what these figures are indicating is that microbial composition varies across body sites. So maybe this seems obvious, but at the time we weren't sure, but it is indeed the case that the microbes differ from one body site to another. So the bacteria that you have in your throat are different than the bacteria that you have in your nose or in your stool, and each body site has a particular ratio of bacteria that exist there, and it's true to the point that it's pretty predictive. If you were to take a sample from someone's skin, but not, if you were to receive it blind and not know where it was from, you could just sample that, or you could sequence that, and you could say pretty confidently this is from the skin and not from the stool or from the throat. So they're pretty unique to their specific body sites. This slide has a few figures, but maybe we'll focus on just A, B, and C. A describes the within sample diversity. So this is the diversity, that's shown within the particular person across their body sites. So again, some sites are more diverse than others. Some sites have many different organisms that live there. Other sites have fewer. And then, but not only is it the case that there's within sample diversity, that there's a different amount of diversity in your throat compared to your stool, there's also a between sample diversity. So looking at comparing one person's throat microbiome to another person's, or one person's stool microbiome to another person's, turns out that that's drastically different too. So it's maybe not as common as we might have thought going into it. And it's also not, the D talks about how it's also not the same over time, that these things fluctuate. They fluctuate from within a person, between people, and in the same person from one time point to another. But again, these different bacterial communities tend to cluster together. If you cluster them together, you see that the oral samples tend to cluster together. The urogenital samples are here, the gastrointestinal samples. The nasal samples have a little bit of an overlap with some of the other groups, but they largely cluster apart from each other, and the skin samples tend to cluster with other skin samples. So it's reliable person to person, relatively reliable what the composition will be. Now this is something that's interesting, and that maybe goes against what we've been discussing, that yes, in A we see sort of the phyla breakdown across different body sites. And as we've discussed, so sort of each of these columns is a particular person, and then the ratio of the different phyla in there. So maybe we can look at the stool, for example. In most people, it seems like Bacteroidetes is the dominant. It takes up most of the area, this blue portion. But in some patients, they have more proteobacteria, or it looks like some of them have this pinkish Verrucomicrobia particular phylum. And so there's diversity between patients, and this is true across the body sites. But although there's diversity between patients and between body sites, it turns out that the pathways that these microbes form a part of are pretty consistent across body sites. So no matter the body site, no matter the person, it seems like the microbes engage in very similar metabolic activity. So that was something that was maybe unexpected. So microbial taxa vary across body compartments, but metabolic pathways remain pretty stable. So all of this is interesting, but it doesn't mean too much unless it means something clinically, or it means something for human health. And it turns out that it does, and that these the taxa that are present in a sample and their function correlate with clinical metadata. So in this case, you have some of these taxa, and these taxa differ across races. The proportion of the taxa, the abundance of the taxa differ across races. It turns out that some of the presence of bacteria can correlate with things like the vaginal pH, with age, with BMI, and so it's interesting that, and it was good to know that these things can correlate with actual phenotypic or actual metadata things that we might be interested in. This is another figure that's pretty dense. I'd encourage you to look into it some more if you have some time, because we can't go over most of it. But it's pretty interesting that this is sort of a phylogeny of all these different bacteria that exist, and they exist across different body sites. You know, we have there around the edge. It tells you what these. It picks a few of these species that might be of special interest. The stars are these organisms which are pathogenic, whereas the circle ones are commensal. And it just goes to show that there's just a wide variety of organisms that exist across body sites. They play different roles. Some of them are pathogenic and cause disease. Others are maybe good for you, but there's certainly just a dense amount of information in the microbiomes across our different body sites. A newer project after the Human Microbiome Project was the Integrative Human Microbiome Project, or IHMP. The Integrative Human Microbiome Project happened between 2013 and 2016 and was mainly focused in microbiome dynamics. It consisted of 3 longitudinal studies, which we'll talk about briefly here soon, and was focused on not only how microbes impact their host, but how the hosts respond to the microbes, so sort of this bidirectional relationship. And it didn't only use genomic information, but also incorporated transcriptomic, proteomic, and metabolomic data. And so I won't get into the results too much, but they helped profile what role microbes play in pregnancy and preterm birth, in inflammatory bowel disease, and in type 2 diabetes. And so there's a lot of information now available for these particular either diseases or the particular state, and it's helped us understand sort of what's going on. There's a special interest in inflammatory bowel disease, where they were able to discover some trends in this disease that are correlated with bacterial abundance, and so that's especially interesting and a potential target for clinical applications. The last major study that we're going to talk about is the MetaHit project, which focused on the human gut. And this sort of especially gets into the question of what a healthy gut microbiome looks like. It was an Illumina-based study where in Europe, they took 124 individuals and characterized 3.3 million microbial genes from fecal samples from these individuals. And it turns out that if you take sort of the superset of the bacteria belonging to these individuals, so if you were to take all of the bacteria that exist, even once in any of them, you end up with 1,000 to 1,150 bacterial species, but each individual only has about 160 of those. So there's a wide, variety. There's a wide the diversity between individuals, and they were able to define both the minimal gut metagenome and the minimal gut bacterial genome. The former being the functions that are present in all individuals. So, what sort of functions are these microbes playing in a person, and what is sort of the minimum number of functions that need to be played for the person to be healthy, and then what are the minimal functions that a bacteria has to have in order to be a bacterium and to live. And so this schematic is really dense, but it sort of depicts the pathways that are involved in a minimal gut metagenome. So what role all of these different organisms are playing in the pathways involved in metabolism of the host. And so that's especially interesting, and it's as you can see, it's a pretty intricate set of things that are going on that these microbes are performing for us. So that gets us into the question of what a healthy gut microbiome is. And maybe in some respects that's an outdated idea. A healthy microbiome isn't defined by a single community composition. It doesn't mean that every healthy person has these 100 bacterial species and no more, no less. If you have more, then you're sick. If you have less, then you're sick. That is not true. It turns out that a healthy microbiome is probably best defined by one that is more resistant and resilient to disruption. So it's able to carry on day to day without too much change. Certain microbial representation may increase susceptibility to infection and disease. We've talked about this briefly, where there's particular bacterial species that, if they're present, it's almost a guarantee that you're not going to be feeling well, or there's going to be something wrong with you, you're going to be ill. And there's certain ones that if you're lacking them, you're probably going to be ill as well. This has a natural corollary in genomic science, if you know anything about genomics, where in genomics there are some diseases which are attributable to a single gene, where if you have the mutation, then you are you're ill, you're sick, you have a disease. Whereas if you don't have that mutation, then you're healthy. That sort of has a corollary, like I said, in gut microbiome science, where in some case, if you have a particular bacterium, you are probably sick, and if you don't have that bacterium, you are not sick. But one of the interesting things, and one of the difficult things, is that it's hard to know if microbial dysbiosis is a cause or a consequence of disease. So does a person with inflammatory bowel disease have this bacterium because the bacterium is causing the disease, or is the disease causing the bacterium to be there? It's unclear which one is causing the other. So that's an especially difficult conundrum. To continue with what I mentioned in the last slide in terms of what a healthy microbiome really is, in the last slide we discussed that it was ecological stability, but you can also define it that maybe this is kind of it's kind of debatable, but maybe there is some sort of idealized composition where maybe this is the prototypical microbiome, or this is the perfect microbiome, or it's hard to describe, but maybe there's this ideal set of bacteria. Or maybe it's just a functional profile, like it says in point 3, that maybe it's not necessarily just a set of bacteria, but what sort of roles are these bacteria playing? And so maybe you don't have exactly the same components, but as long as it's doing the same job, maybe that could be considered healthy too. So all of these considerations were used, and I'm gonna plug something that our lab is involved in, Dr. Mazumder's lab at George Washington University. Is the creation of this gut feeling knowledge base that we developed, and it's a sort of a reference list of these microbes that exist in the gut, and or at least in the healthy gut, and what abundance profile these microbes have. The hope is that it has sort of a clinical applicability, and that it's sort of this baseline grouping of organisms that can be used as a control against dysbiosis. So here's what a healthy microbiome looks like. Compare, your samples to this healthy set of microbes. And one of the big things is that this was created from, we took samples, bacterial samples, from stool samples, and sequenced them and put them analyze them against a filtered database that doesn't have artificial sequences or environmental sequences or lab strains, but that has only bacteria that you would expect to see in a human gut. And it was further subjected to some manual curation and some additional computational steps that led to a final product that we were able to say with high confidence constituted real reads that were observed, and that really sort of this profile that really does correspond to a healthy gut microbiome. So we've talked thus far about the history of of gut microbiome science, all the way back from Leeuwenhoek and the origin of the microscope and studying bacteria. We talked about germ-free animals. We talked about some of the early studies of sort of what applicability these things had. We talked about some of the analytical methods that we use, and now most recently we started talking about what some of these big studies that have led us in the direction of understanding what a healthy gut microbiome is. And now let's sort of get into the applications. We've been hinting about this all along and talked about it briefly, but let's talk about some of these applications that gut microbiome science might have, especially in a clinical setting. So the gut microbiome is implicated in diseases like obesity, diabetes, autoimmune disorders, metabolic disorders, cancer, and neurological disorders. It has some clinical applications in developing therapeutics, in making some diagnostic tools or conducting clinical trials, and there's a variety of commercial products that could be developed due to these findings, like probiotics, prebiotics, medical foods, supplements, and I'm sure there's a host of other things that will be developed in the future. Maybe some pitfalls to consider. We've talked. There's a lot of caveats in microbiome science, and translational gut research isn't immune from these same caveats. It's important to know that an individual's microbiome isn't as stable as you might think. It can change from day to day or from testing period to testing period. Diet doesn't always have the magnitude or the speed that we might expect it to have in microbiome composition. Again, there's a high degree of diversity between people, so not everyone has the same stool microbiome. And very importantly, different laboratory and computational methods will yield different analytical results. So, if one researcher is using a particular DNA extraction method and a particular computational pipeline, and another researcher is using something that's different, they might arrive at two different results, even though their samples might be very similar. Keeping that in mind, there have been some really important clinical advances in the past 15 years, and so this is just sort of a general list of what those are. We've gained the ability to look at a strain level resolution of these microbes, meaning that we can see with even more definition than even a species level. So just like dogs are all the same species, but a Great Dane and a Chihuahua are a little bit different, microbes have sort of the same thing going on, where a particular species of microbe might have two separate strains, one of which is maybe pathogenic and the other is not. So allowing us to have a strain level resolution is really important, and that's an important advance in the last 15 years. Mass spectrometry has been really important in looking at proteomics and metabolomics as they relate to the gut microbiome and discovering bioactive compounds. We talked about earlier the transfer of microbiota in these fecal microbiota transfers between the humans and the mice. Well, that could happen between person and person, and it's been done successfully. There's a lot of clinical trials that you could transfer fecal matter from one person to another, and that could be really beneficial in restoring a healthy gut microbiome. There's a lot of emphasis in early life microbiome health, and how your microbiome starts sort of even in the womb, and obviously as you're born, and there's a difference between babies that are delivered vaginally versus delivered via C-section, and how those differences might have long-lasting consequences. There's been some studies into dysbiosis, especially in IBD and irritable bowel syndrome and other diseases related to the gut, and the role that microbes play in those diseases. There's also been recognition of specific microorganisms as pathobionts. So these are organisms where pretty much if they're found in your gut, you're going to have something wrong, or if they're not found in your gut, you're going to have something wrong. These have been implicated in colorectal cancer, in inflammatory bowel disease, in diabetes and obesity, in Crohn's disease, and I'm sure there are more still forthcoming. There's also interest in the role of diet and the gut microbiome, especially a Western diet and sort of people who migrate to industrialized countries and the changes they might see in their microbiome. Also, the role of food additives or urbanization, countries especially that are undergoing industrialization or urbanization, and how those changes can have irreversible effects on the gut microbiome. And like I've mentioned several times, FMT, fecal microbiota transplantation, that's been an especially interesting field, and there's a lot of clinical trials that have been registered to do that. And finally, let's start to get into the realm of future research and what is to come. Something that's especially interesting and something that we've noticed in our group as well is this microbial dark matter. So kind of like how your genome has this junk DNA, which is probably not junk, but this part of the genome that we don't really understand. The same is true of a microbiome sample, that often there are organisms within that we don't know what they are. We know that they have genomic material, but are they fungi? Are they viruses? Are they archaea? And maybe these organisms play a more important role than we thought. Maybe it's not just, throughout this presentation when I've talked about microorganisms, I've mainly focused on bacteria, but perhaps other kingdoms play an important role as well. There's, as I sort of mentioned briefly before, there's an emphasis on early life interventions, things that you can do maybe in the womb or shortly after for an infant that could influence their healthy microbiome and immune development. Hopefully, as we continue to collect more and more data, we'll be able to create predictive models of microbiome function, and not only have it be a European data set or an American data set, but hopefully this is globally represented. People across the world have different genomes, slightly different genomes. They have different diets. They have different environment. So all that can play a role. So maybe what we discover in one population might not be true of another. And so as we continue to collect more and more data, hopefully that allows us to have some more predictive power and to get a scope of what exactly human health really entails in terms of the gut microbiome. There's also a big focus in the role of diet, as I briefly mentioned before. But if you can discover, foods that lead to a healthy microbiome or have desirable features, maybe there could be genetic engineering to make these plants have those characteristics, so that people, maybe in developing countries or people in even developed countries, so that they can eat healthier foods that help encourage a healthy microbiome to reduce the incidence of gut-related diseases and diseases of all types. So all of these things are currently happening in the field of gut microbiome research. If you have any questions, feel free to reach out. I hope that this has been helpful. Again, we sort of explored how we got to this point in gut microbiome science, the history behind what's happened, some of the major studies and some of the analytical tools that are being used, how it's possible to do some of these things. And at the end there, we talked about the clinical applications, what's currently being developed, and maybe what's in store for us in the future. So thanks for listening.